this afternoon, our co-host is Annalise Tesley. She is a biochemistry junior pre-med from Westmont, Illinois. And Annalise, what is your career plans? What are your career plans?
Right now I'm pre-med and I'm not sure which area I would like to go into, but I do have an interest in dermatology.
Oh, okay, okay.
Yeah.
It's very nice. And I'm assuming you went to school in the Illinois Westmont area or something?
Yes, I did. Yeah.
Okay, very good. What are some of your hobbies?
Well, I play the oboe. I've been playing for 8 years, so I'd say that's my main hobby, probably.
Okay, very nice. Well, this afternoon, Annalise will be our co-host and would go ahead and introduce our speaker for today.
Thank you for the introduction, Dr. Murray. Hello, everyone. Thank you all for joining today's chemistry seminar. Today, I have the privilege of introducing our speaker for today, Professor Salwa Karboune. I hope I am pronouncing that correctly. I'm probably not.
No, it's good. You're doing it well. Yeah.
Okay, thank you. Professor Karboune is the Associate Dean of Research at the Faculty of Agricultural and Environmental Sciences at McGill University. Her main research expertise is in the design and development of new green approaches to synthesizing novel functional food ingredients that can help improve food products' quality and functionality. Her multidisciplinary research program bridges the gap between food chemistry and enzyme technology, and additionally provides access to new, well-defined food ingredients that cannot be obtained by conventional methods. Her expertise extends to the progression of integrated approaches in the development of new food products and extending the shelf life of food products. Her research program has garnered national and international recognition. Her research program has been presented at worldwide known conferences and published in more than 1,200 peer-reviewed journal articles. She currently leads the Consortium for Research Innovation and Transformation of Agrifood at McGill University, which aims to promote innovation in the agri-food sector and deploy effective solutions to produce safe and nutritious food from flourishing, managed natural food systems. Professor Karboune is passionate about science and active learning, and she places a high value on creation, which manifests through her innovation and her drive to translate knowledge into real practices. So without further ado, please join me in welcoming Professor Karboune.
Okay, thank you. Thank you very much, Annalise. A very nice introduction. I'd like also to thank Dr. Murray for giving me the opportunity to speak at your class. McGill is located in Montreal, in Quebec, in Canada, and we do have a few faculties and schools, and Faculty of Agriculture and Environmental Sciences is one of the faculties where I am located. So if you have been in Montreal, so Montreal is an island, and we do have 2 campuses. So we have one in downtown where we have all faculties, except the Faculty of Agriculture and Environmental Science, which we do have it at the end of the west of the island. And that's what we call McDonald campus. So we have the main downtown campus and we have the McDonald one. We have a farm, of course, at the Faculty of Agriculture, but also we have a School of Nutrition. We have also a Department of Bioresource Engineering, and all that help us really to bridge the gaps between the different disciplines. This is a nice picture of our farm, of our campus. So that's certainly not as of today because of the winter, it's very snowy, but in the summer it's a very nice campus, and there is a lot of activities that are being happening in our campus across the year. Okay, So I'm here today to talk about what I do as research, and also I would like to spend sometimes why I'm doing this research and what is the motivation behind it, and what is the impact that I'm looking to make through different research activities that I'm doing. So of course my research is related to food. We know how food is important. We know how food is beyond providing as nutrients. It does also provide a social environment, happiness. So it's really very, very important in our life. And every day we need to make decisions about what we eat, to buy, to prepare, to consume. And those decisions are personal, social, and emotional, and they're very important decisions. Now, it's not new. It's a trend. We know that the consumers are beginning to associate healthy diets as a cheaper way than going to the doctor. The interaction or the link between nutrition, food, it's now much more going closer. So in a way to prevent many chronic diseases, to live longer, to not go to the doctor, it's just trying to get a good, healthy diet. And that has been one of the important endeavors that I do believe into it, and that also I'm looking to contribute to it as well. So I like a lot this saying from Charles Darwin that he mentioned, it is not the strongest of the species that survives, nor the most intelligent one, but the one that is more responsive to the changes. So we know that our society, what we eat, pandemic has been a very, very, very good, I would say, unfortunate circumstance that help us to increase our ability to adapt, because we had to adapt. Our agri-food chain has to adapt what we eat, what we are able to produce, what we are able, as well as society, to take to pursue. So if we look at the trends in the agri-food industry, from where I'm very active, we see a shift. And of course, as I mentioned, this shift and focus on priorities, it's not new. It has been happening for many, 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 many years. So through the years, we're trying to move from a product and crop-driven platform, where most of the products that we consume, we produce, we promote as a society, they have been merely product and crop driven. Now we are more in the function driven platform, where we are looking to the food, what can provide me as a functionality. Do I have a food which is going to provide me proteins, fiber, will prevent some of the chronic diseases, and so on. And I personally do see this as shift as an opportunity for us as being active in the agri-food sector, to become the leader for nutrient deficiency management and disease prevention. Like the pharmaceutical company, they are the leader for disease management. So that's an opportunity to see how can we make the food beyond just providing nutrition, but to help us as well to prevent some of the chronic diseases. So as I mentioned, the function-driven platform, it's not something new. It has been there for many, many years. And the first generation that we have, it was food fortified. So up to date, we have many of our food products that they are being fortified with iron, fortified with calcium, fortified with that, because that has been the case. So we were looking to the fortification. We moved to the second generation, where now we can have some food that they have some claim, and that I'm talking about functionality claim. So food enriched with omega-3, food enriched with probiotics, prebiotics, all those foods are functional because they provide an advantage, they provide a functionality, and we consider them a second generation. And we are working toward the third generation, which we are looking to the multiple benefits. And then we have the future generation, which is the nutrigenomics. So let's look at each one of these platforms and what they do have as a difference, and how can we, and where we are going. So where we are today and what we are working on it for the next 10 years and where we are going in the future. So as I mentioned, the fortified food, they have been there to prevent some nutrient deficiencies, and that was through supplementations. So when we do make functional food with the second benefit, this time we fortify with the bioactive molecule that can improve the health, prevent the chronic diseases. But as we are understanding how these bioactive molecules are being metabolized, and understanding as well that there is a lot of synergistic interaction between these bioactive molecules, we're moving to the third generation that I'm working on it, is to try to develop a food product, a functional ingredient that will provide the multiple health benefits. And to sometimes, in many cases, those bioactive molecules do exist in our current food, but their bioavailability is very limited. How can I increase the bioavailability? Because at the end, when we are consuming them, metabolizing them, we need to be able to make them bioavailable. So you have certainly heard about the nutrigenomics, where we are trying to, based on the genetics of each individual, to tailor a diet to personal genetics. And we know how genetics can be very complicated, but nutrigenomics technology, I have some colleagues working on that, is from the genetics of each consumer, we can predict what are the most likely disease that he may get. And based on that, we can tailor the diet in a way to prevent this disease from occurring. So in the future, by promoting this nutrigenomics food, we will have a shift of market from being more like a system pool to a consumer pool system, because it's going to be by individual needs. And of course, we are still not there for the time being. So what we do as research, so my lab, my group, what they do as a research, we develop innovative strategies for the production of functional ingredients and nutraceuticals. So we have different strategies that we have been taken in diverse projects. The first one is designing a new molecule. So how can we design a new molecule? As we understand the biochemical function and the functional properties of many bioactive molecule, we know which molecule and what are the chemical characteristics that this molecule should have to be able to metabolize in an optimal way. So I believe strongly that the chemistry is the ground of any bioactivity. And if I am able to control the chemistry of my new molecules, I will be able to also modulate the biochemical function in an optimal way. We are also creating value. So we know that many bioactive molecules, if we combine them together, they may provide a synergistic effect. They can provide even higher value than if we consume each bioactive molecule separately. And in some cases, we have a long polymer. Just controlling them, hydrolyzing this polymer into oligomer, it can provide also some health benefits. Improving functionality and bioavailability. So it can happen that some bioactive molecules, they do have a health benefits, but they are not bioavailable. We need to change their physical chemical properties. So this can also be achieved through some strategies that can be used to improve the physical chemical properties that can lead to an improved functionality and bioavailability. And the last strategy has been, in some cases, we were able to identify some good bioactive molecule, but in the nature, they exist in very low amount, or it's very difficult to isolate them from a food system. So we try to find a way to maximize their synthesis. So to be able to achieve this strategy, so of course, I'm using biocatalysis enzyme technology at the center. I will explain later on why I'm using enzyme and why I believe strongly in the ability of the biocatalysis. But I'm working at the interface between food chemistry and the functional food science, where functionality of the food is beyond just having nutrients. It does provide and help to prevent some chronic diseases. So biocatalysis, which is a subfield of biotechnology, it's a field where we use enzymes. So enzymes do exist naturally. We have them in our body. They play an important role. They are considered as a biocatalyst. They are natural biocatalysts. Now, when we produce any functional ingredient using a biocatalytical approach, it can be labeled as a natural because I'm not using chemical or I'm not using any toxicant. So that labeling is so important. We can also obtain them with the high quality, and that's because enzymes can provide a high specificity and selectivity. So what I mean by that is that the enzyme, because of their structures, their proteins, they can be specific and selective to which substrate they can convert, and they can convert the substrate to a specific bioactive molecule, and they can also be selective to which reaction to catalyze. And this specificity and selectivity does provide some advantage if we compare them to the chemical pathways, and that help us to obtain a high quality food ingredients. I put here the cost, but it depends on the enzyme, so that's important to keep in mind. So some enzymes that they have been developed, we're able to produce them efficiently. The process can be cheap, but if we are dealing with enzymes that they're still under development, we still need to express them very well to produce them, the cost may be higher. And of course, it's a green process. So it's a green process, it's an environmentally friendly process. So the challenges, of course, the enzymatic synthesis does provide a lot of advantages, but there are also some challenges that we need to be aware of when we are trying to use an enzymatic process. First of all, is we need to, the type of biocatalyst is so important. And even the same type of enzyme from different microbial sources, they may not have the same structure, they may not lead to the same results. So the type of catalyst, the source of biocatalyst is extremely important. And also enzyme, the fact that they are very thermolabile and we need to control their active site where the substrate comes and binds to them. So we need to be able to modulate the microenvironment of the enzyme, like surrounding the enzyme, and even the microenvironment of the enzyme, because the microenvironment and the microenvironment can affect the three-dimensional structure of the enzyme, and that at the end can affect our yield, our specificity, selectivity, and so on. So it added more, I would say, more requirements of really putting them under control conditions that we can get the product that we're looking for. There is also another important aspect, which is the stability, because some enzymes, they have different stability. And here we're talking thermal stability at different temperature, because they can be denatured. And when they are denatured, they cannot function very well. And even we have stability, they can be inhibited by some end product. So if you have a reaction and you're producing an end product, which is not your main product, but maybe just a bioproduct, it can also denaturate the enzyme. So that is something to keep in mind. It's the stability, the activity, and the microenvironment to be able to use them efficiently. So what I'm going to do for the next, I would say half an hour, I'm going to provide you an example of projects that I did work on it and in which I use enzymes. So the first project, it was nearly linked to design a new molecule. As you know, prebiotics, in our colon, we have 1,000 and 1,000 of bacteria. And we have a good bacteria and we have a bad bacteria. So the good bacteria, they can help they can stimulate our human function, they can inhibit the harmful one, they can help with the digestion, they can help with the absorption of vitamin, mineral, they produce a good short-chain fatty acids. And the microbiota is like now it's really well studied, and there is still a lot to learn as we are linking it to our overall health. I personally think that our microbiota is the brain of our health. So which means many things happening on our microbiota, it helps us to see how the link to other diseases. So it's not an indirect way, it's most of the time an indirect way, but where we have learned a lot and there is still a lot to learn. And we have the pathogenic bacteria, of course, that they can lead to many disease, infection, cancer, and so on. Now, we use prebiotics that they stimulate the growth of the beneficial bacteria, while they limit the growth of the harmful bacteria. Fructooligosaccharides, they are widely established in the market. We're using them for some type of food product. So we can produce them from inulin, from the artichoke, and so on. But the existing one, they do suffer from some limitation. And what we are doing is that we identify those limitations, and we're trying to see by modulating the chemistry of these prebiotics, can we enhance their properties? Can we improve them? The first one is the selectivity. Remember, in our microbiota, we have a good bacteria, we have a bad bacteria, right? So I want my prebiotics to be mainly used by the good bacteria. And that's where the selectivity is very important, is to have this bacteria, the ability to metabolize the prebiotics, my fructooligosaccharides, but not the bad bacteria or the normal bacteria. So for that, there are some studies that show that when you have fructooligosaccharides with the beta-2,6 linkage, they get higher selectivity than you have beta-1,2 linkage. And the one that they do exist in the market, currently, they are with beta-1,2 linkage, 2,1 linkage. So the idea was, can I synthesize this fructooligosaccharides with beta-2,6 linkage? So can I increase my selectivity? The other challenge here is the fact that most of the existing fructooligosaccharides, they are metabolized at the proximal region of the colon. Well, most of the disease are originated from the distal region of the colon. So how can I increase the colonic persistence? So basically, I can increase the colonic persistence by increasing the length of my fructooligosaccharides, but not too much that I will lose selectivity. So it's really looking to the high molecular weight fructooligosaccharides longer, then they can reach the distal region, and they can be used by the good bacteria as a carbon source in the distal region of the colon. Now, we know that the harmful bacteria tends to absorb on the epithelium cell of the colon. And we know as well that having some fructooligosaccharides headed with galactose, with xylose, they may help to improve the anti-adhesive properties. So the idea here, can I have my fructooligosaccharides headed with galactose instead of having it headed with the glucose, and would that make a difference? Of course, here it's really from the understanding of how they are metabolized, we're trying to come with solution, chemistry from a chemical perspective, and try to find a way how we can produce those molecules. So moving on to the next, so we have identified one enzyme, which we call it 11-sucrase enzyme. And what is the advantage of 11-sucrase enzyme, which is a fructosyl transferase enzyme? It is part of a, when we talk about enzymes, there is many families of the enzyme, and each family, they have a specific action, they have specific feature in common between them. So 11-sucrase is part of the glycosyl hydrolase family. And we can look at also the structure of this enzyme, which is being characterized by how many from a secondary or tertiary structure, they can have a specific feature as well. Now, 11-sucrase is a very potential enzyme because it can provide us a diversification of the reactions that can catalyze using only sucrose. So I just take my sucrose, white sugar that you eat each day, and I add my 11-sucrase, and many reactions can happen. So why this enzyme is very unique? Because it can use the energy of cleavage of sucrose, because sucrose is made of glucose and fructose. And when you have this enzyme, it will use this free energy to transfer the fructose to another molecule. So I have a donor, which is the sucrose, will donate the fructose, and I have another molecule, which is a fructose, which is going to accept my fructose, and it will accept it with the beta-2,6 linkage. If you do recall, this is the linkage that I'm looking for. Okay, So now we have been working on 11 sucrase for more than 20 years now. And we were interested first to know how, depending on their microbial sources, we are isolating, we're producing them, they are being extracted from Xenomonas or from Bacillus aeruginosa. They don't function in the same way. Some leaven sucrase will only produce fructooligosaccharides and oligoleaven, short chain, medium chain. Other will produce the leaven, which can be very, very long polymers. And we wanted to understand why some they do that and the other more oligomerization, other they do more polymerization. And there is here a process that has been published that explains that, because you have to imagine in your enzyme, you have many subsites, right? And the subsite, they can hold the growing chain of your oligomer as you are adding one fructose. And depending on this subsite, you can get either oligomers or you can get polymers. Okay, so we use only sucrose. Now we said, okay, what will happen if I use a sucrose analogs? As you know, sucrose, which is your white sugar that you eat each day, it is made of glucose and fructose. So we made these sucrose analogs by replacing the glucose with galactose, with xylose. Can still the enzyme function? Remember my targeted fructooligosaccharides, I wanted a mixed fructooligosaccharides with different heads. And I can see from here, then when I put, when I'm using sucrose analogs, I can have the head different from the glucose as I was targeting. So we were very nice and nicely happy to see and glad that my 11 sucrase can use also the sucrose analog as a substrate, which they provide me with the opportunity to be able to produce this mixed fructooligosaccharides. Now, when I take sucrose and I add another acceptor, like for instance, I put sucrose plus maltose and I add my enzyme, and here I found out that my enzyme is also able to transfer the fructose from sucrose to maltose, and to get, for instance, here what I call the L-rose with a quite good yield. If you look at this L-rose here, it's a trisaccharide that I can find in honey in very low amounts in honey, because it can provide, it's a prebiotics, it has some immunostimulatory effects and so on. But now with my enzymatic process, I'm able to produce it in a high amount by using 11 sucrase. So that's where the specificity, the selectivity, it does really broaden the application of the molecule that I can produce. I did the same thing with raffinose. So can the enzyme transfer the fructose to raffinose? Yes, with the yield of 55. Can my enzyme transfer the fructose from sucrose to lactose? Yes, and I'm able to produce the lactose sucrose. And this is very interesting. I don't know if you know, but lactose, we can find lactose in milk, and lactose, many people, they have intolerance to lactose, right? If I'm able to convert the lactose into lactose sucrose, I may be able to decrease this interference because I'm adding another fructosyl unit to it. And that can open new avenues. Now, the specificity of the enzyme is an important feature, but each enzyme, depending on from which microbial sources I'm getting it, I can get different specificity. So for instance, here, if you look at it, from Bacillus aeruginosa, it shows a very broad specificity here, but Bacillus subtilis has some specificity as well. That's why the selection of the microbial source of your enzyme, it's important when we want to use it for some applications. Okay. So we have developed the process. We were happy with it. We did many publications, and then we said, okay, what will happen if I take my enzyme and add it to maple syrup? Of course, you know, maple syrup is, it's not only in Canada, in the United States as well, but we are in Quebec. They are the most important Canadian producers of maple syrup. And why maple? Because maple is made of 90% of sucrose. And remember, this enzyme likes sucrose and converts sucrose into oligomers and into fructooligosaccharides, into leaven, and which is a big polymer. So that's what we did. we took the maple syrup with different bricks and we added by enzyme, and then we looked at what are different units of the enzyme, different temperature, and we tried to see which if the sucrose was converted, and it was converted to what. So we find out, I'm just giving you the optimization, that depending on the bricks, right? When I have 140 gram per liter of bricks of sucrose, I'm able to produce mainly fructooligosaccharides and oligolevain, okay? When I'm using a bricks of 66, I'm able to enrich it with oligolevain and levain. So imagine now you can eat a maple syrup enriched with the prebiotics, because I'm able to convert some of the sucrose into prebiotics, okay? And that did make some headlines, so showing how an interdisciplinary science here at McGill, where we were able to enrich the maple syrup to become, well, I would not say a superfood. I'd never asked the journalists to mention that, but that was mentioned here. Okay, my other example that I want about it is how to create a value and how to improve the bioavailability. So creating a value and bioavailability. So as you know, so we have quite strong interest in my lab in prebiotics, right? But at the same time, we the antioxidant activity does interest us as well. And there is some carbohydrates that they are phenolated. It means they have a non-digestible carbohydrate, can be oligosaccharide, can be polysaccharide, and they are linked through an ester linkage to a phenolic compounds. We can find this ferulated carbohydrate in some in sugar beet pulp, for instance. If you take the sugar beet pulp, you can have this ferulated carbohydrate, but when you extract them from the sugar beet pulp, the yield is very low. Like we are not more than 5% yield. So they are very interested molecule. There have been some study, animal study, showing their potential, but we cannot really take advantage of them because it's difficult because the yield is limited. Now, why we are interested in this molecule? We're interested in the molecule because they have double functionality, which is prebiotic and antioxidants. And at the same time, if it's like I'm taking an antioxidant and attaching it to something which is non-digestible. So it's like I'm using it as a carrier. I'm using my carbohydrate, non-digestible carbohydrate as a carrier to carry the antioxidant activity until my colon. And it's another potential advantage instead of only having the phenolic compounds. So we had to look at a way of how to synthesize these ferulated oligosaccharides. The challenge was, of course, to select the appropriate biocatalyst. Now, what has been a big challenge here is that we are dealing with two substrates, carbohydrates and phenolic compounds. They don't have both the same physicochemical properties. Phenolic compounds are mainly soluble in hydrophobic media, and I have my carbohydrate, they are mainly soluble in water. So which media should I use to put them together and come close together? So we use microemulsion, nanoemulsion to do that, to be able really to have a media where both substrates are available to the enzyme. And we had to modulate the reaction parameter as well. So we use the enzyme ferulyl esterase, and we use a nanoemulsion and microemulsion to be able to use them. So when we started doing this, developing this biocatalytic system, we used different substrates. We use arabinobios, xylobios, galactobios, sucrose, raffinose, xyloligosaccharides, and fructooligosaccharides. Now, when we look at the yield here, you will see that the bioconversion yield is very, very limited, right? I have the maximum 11 and 13.2. So we thought, okay, that's not really what we had expected. We wanted to have higher yield of conversion, then it can provide that it can increase the efficiency of my biocatalytic process. But when we did the MS analysis, we realized that some carbohydrates are not only ferulated with one ferulic group, but I can have 2, I can have 4, and it's it's a kind that I am underestimating my yield, because my yield is based on how many carbohydrates has been phenylated. But in the reality is that one carbohydrate can be conjugated or linked or esterified with more than one ferulic. And then, of course, we did the antioxidant activity, because we want to make sure that the antioxidant activity of the ferulated components will not be affected if they are just ferulic acid, or they have been esterified with the carbohydrates. And we find out that there is not significant change. And in some cases, we have a little even enhancement in the scavenging ability of those components. So after developing the process, we had one project where we have been working on cocoa bean shell. So the cocoa bean shell is a bioproduct that we recover when we make chocolate. And for the time being, it has no value. So our idea, it was, okay, can we extract oligo and polysaccharide from the cocoa bean shell, and then after that ferulate this cocoa bean shell to get the sweetener prebiotics with some antioxidants. So it's really trying sustainably to use a bioproduct as a source of our polysaccharide and do ferulation. So basically, so just to show you some picture of some cacao bean shell. So there is more than 7,000 tons that are discarded annually. Each time we make a chocolate, we have this shell that's being discarded. So we were interested in first of all, analyze it, do some chemical profiling, understand the polysaccharide that we can find in the cacao bean shell and so on. And then from that, we were able to produce a cell wall material from the cacao bean shell, fermented, dried, or roasted. And we extracted the polysaccharide. We use a multi-enzymatic product to generate some oligosaccharide, and we use this oligosaccharide to ferulate them. So it has been a long process to go step by step to be able to produce functional ingredient that we can put back in the chocolate from a bioproduct of the chocolate. And of course, the fact that we're using some oligo and even short chain carbohydrate, they can provide some sweetening ability. So instead of using sugar, our idea was, can I produce sugar from the bioproducts of the cacao bean shell? So if I focus on the ferulation, as I mentioned, so we did the ferulation. We used different variety of the cacao bean, and we did ferulation of the polysaccharide, of the oligosaccharide, using two types of ferulasterase. So the result shows that we're able to get a good ferulation. So we were happy with that. And then after that, we looked at the antioxidant activity. So we can see here, if I use my polysaccharide from the cacao bean shell without ferulation, my antioxidant activity is low. But when I do ferulation, I increase my antioxidant activity. And even in some cases, if I compare it to the ascorbic acid, to the ferulic acid, I'm getting a high antioxidant activity than the ferulic acid and the ascorbic acid. We were also interested in, if I'm producing this ferulated oligosaccharide and polysaccharide from the cacao bean shell, I want to know their prebiotic index. The prebiotic index gives us an indication how much they can stimulate the good bacterial growth. And of course, we compare that with existing inulin, which is well established in the market, to see if it is comparable or it has a higher prebiotic index than the inulin. So we use two types of lactic acid bacteria, and we looked at how the prebiotic index vary over time using the oligosaccharide from the cacao bean shell and using the rhamnogalacturonate as an example, as a standard, and inulin. So we could see from this result that our oligosaccharide from the cacao bean shell provide a comparable prebiotic activity to the inulin and rhamnogalacturonate index. But you can see here in the case of, for this strain, that we have a kind of activation in the prebiotic index as compared to the other standards. Of course, when you stimulate the growth of the good bacteria, you do expect to have the production of short-chain fatty acids, because that's how can we measure the activity. And that's what we did. We looked at how much of the short-chain fatty acid we are producing. And we could see from this result that the short-chain fatty acid that were produced because of the prebiotic index and high activity was lactic acid and acetic acid. So that gives us more confidence about the ability of this oligosaccharide from the cacao bean shell to stimulate the growth of the good bacteria. Okay, maybe just another example. Even though I plan to give 2 examples, I would just focus on one, which is about creating value and maximizing the synthesis. So the potato. So potato, very abundant, right? And it has been associated a lot with bad diets, right? Because of the starch contents. But we were interested in this study in other components of the potato. We were interested in the potato proteins. We were interested in the potato pectic polysaccharide. And we were finding a way, how can we combine the potato pectic polysaccharide with the potato proteins? So why potato pectic polysaccharide? Because they can provide some beneficial properties. So the pectic polysaccharide are pectin, but when you talk about pectin, it's a quite complex structure. So we can have a rhamnogalacturonal region, xylogalacturonal region, homogalacturonal region, and a rhamnogalacturonal 2 regions. And depending on the source of this pectic polysaccharide, either we're getting them from apple, from citrus, from sugar beet, you don't get the same chemical characteristics. You don't get the same percentage. Now, what makes potato pectic polysaccharide unique, one important feature that really attracts us is that they have a very high galactose content. And that we were very happy to see that, because when we have high galactose content, it means that I can use them as a source of fiber. I can use them to produce galacto-oligosaccharide, and they can also be used as a prebiotic and so on. So that makes them unique if we compare them as compared to orange, apple, and sugar beet pectin. Now, having galactose-rich rhamnogalacturonane, it can help to prevent the tumor growth, and that's because the galactin comes on the surface of the tumor cell, and they can actually bind them, right, via the galactins, and by binding them, they will actually limit their growth. So the idea here was really to that some of the health benefits, and that also can have a positive effect on the human system, in addition of being a dietary fiber, and in addition of having a prebiotic and anti-adhesive properties. So we had to find a way to produce them. And of course, as I do believe, and I'm very interested in the enzymes. So what we did in this project is that we compared the enzyme and the alkaline process, just doing simple alkaline extractions. So if you look at the yield, you will see, yes, it's comparable if I'm using 1 molar alkaline solution, if I'm using 1 molar sodium hydroxide solution. But later on, if you try to see what you are really isolating, you will see that when you use the alkaline extraction, you recover a lot of fragments. And the reason why is because you are breaking down this component, right? So that's why we thought, okay, maybe what we should do is the alkaline extraction here. So what we did here is that we had used the enzymatic process, but here we see that we don't have debranching, we don't have a defragmentation. And we were able to prove by NMR and by many other techniques that we are able to isolate the rhamnogalacturonal region efficiently and without having any debranching. And that was one of our goals. Now, when we got this rhamnogalacturonal, we wanted to hydrolyze them to get the galacto-oligosaccharide and the arabinogalacto-oligosaccharide. And for that, the use of the enzymes, the fact that they are specific, they have a high selectivity, it can help me to get a very specific product. So we tried different multi-enzymatic products with different units. Of course, we had to limit those that they have low yield. We had to limit those that they generate only non-galactose type of oligosaccharide. And we end up by selecting specific enzyme that was able to produce the galacto-oligosaccharide that I'm targeting. So with this, we had to show that my galacto-oligosaccharide is non-digestible. Remember here, when I need to have a prebiotic activity, it should not be digested and it should reach the colon. So we use a Team 1 system, and that's mimic the stomach, the duodenum, the jejunum, and the ileum, and it helps to determine if it is digestible and non-digestible. So by doing that, we find out that our monosaccharide, our rhamnogalacturonan, is non-digestible because I'm able to recover it after digestion. So I got 79.3% of what did I feed, which is very high, of polysaccharide, and I got to 81.6%. So I'm already providing a proof that my rhamnogalacturonan is non-digestible and will reach the colon. My next question was, now that it's non-digestible, can it stimulate the growth of the good bacteria? So in the other study, we use one cell, one lactic acid bacteria cell, but in the study, we took the fecal matter and we tried to mimic our flora, but we're using a mix of bacteria that we have in our digestive system. And we looked at the bacterial species, we looked at how they change if I feed them with the fructooligosaccharide, if I feed them with the potato rhamnogalacturonan, or with the hydrolyzed one. Now, the good news here is that the Clostridium leptum, which is a harmful group, has been decreased, which is good. And I have an increase in some lactic acid, lactobacillus. I have some increase in lactobacillus, and I think in bifidobacterium. And that shows that I have a prebiotic effect and I have a good selectivity because I'm able to stimulate the growth of beneficial bacteria and to inhibit the growth of the harmful bacteria. And of course, when we talk about prebiotics, I need as well to assess the ability to produce the short chain fatty acid. Because if I have the stimulation of the good bacteria, it means that I will produce enough short chain fatty acid. And that was the case. Like if I compare it to the control, I can see that I'm able to produce high amount of butyrate, and I'm able to produce a high amount of the total short chain fatty acid if I compare it to the control. Okay, I think because of the lack of the time, I'm going to stop here. I was planning to talk about the potato protein, unless Dr. Murray, you advise different. I believe that the time.
Yeah, we could open it up for questions.
So maybe if I'm stopping here, I want just to go and acknowledge my students, make a conclusion rapidly. So there is an increasing interest in the generation of unique ingredient nutraceutical, starting from simple components. And I believe strongly that the biocatalysis, the enzyme technology can help to develop the system that can help us to produce these ingredients. And of course, I would like to acknowledge all my graduate students that they have contributed to all research that I presented today, and all the funding agencies that they provided the funding to be able to do this research. And thank you very much for your attention.
Okay, thank you so much. Yes, very good. Food chemistry, very important. So I got a question to begin things. Is any of your technology and stuff commercialized? You work with companies or you have your own company? Is this going to market?
Yeah. So I work a lot with the food companies. We have patents, a few patents that are being now. Is there any of this process that I mentioned already in the market? I would say none of them for the time being, but we are working. We have patents, and we are trying to go now to the next step in the technology to have them implemented.
Okay. Another question is this, so you do a number of enzyme reactions, right? So what's the scale are we talking about at this point? Is it just benchtop type scale, or have you done any work again with the companies in terms of scale up?
We did scale up some of our processes. Like an example for potato process has been used as a scale up. For cacao bean as well, we did a scale up as well with the. So some of the process has been in a scale up level, not the benchtop level.
Okay. Okay.
Yeah.
Right.
And enzymes are already used in the agri-food industry, right?
Right.
Like,
yeah.
To give you an example, when we want to extract the juice, we use enzyme to open the cell wall and to extract most of the juice, right? Depending on which type of the juice. We use enzymes are very, very important when we want in the bakery industry as well, when we want to make bread and extend the shelf life. So they have a quite wide application. And in some cases, they are being produced during fermentation, and they help the fermentation process as well.
Okay. Anyone has a question? Could unmute your mic, your sound. While people are working on their questions. So sometimes immobilizing enzymes make them become more efficient. Have you guys looked at that process too? Yes,
absolutely. We did. I of course I couldn't cover it, but the enzyme immobilization is an important aspect that we do consider, and we have done few research where we make our own support, we modify the support, because you remember when I mentioned it's important to control the microenvironment of the enzyme, and the only way to control the microenvironment, one of the way, not the only way, one of the way is to immobilize the enzyme on the support that you can able to control its microenvironments. So immobilization, and also it can allow a reusability, because that's how you can decrease the cost of your process.
Right, correct. Anyone? I'm looking to see if there are any hands up. So
I think there is
David, Dr. Randall?
Yeah, so that's very interesting. So you mentioned about the maple syrup, where you can take and add an enzyme, and it changes some of the maple, the sucrose in the maple syrup to a probiotic material. And so I was wondering, how does it affect the taste or texture of the maple syrup? Because you're basically taking sugar away.
Yeah. So we haven't done the sensory evaluation, right? But the texture, there was no significant change in the texture. So and also we can control how much with the sucrose you want to convert it as well, right? If you want to enrich it 20%, there is a way of controlling it. And what we are doing is really converting the sucrose, because the sucrose will be converted into oligomers of fructooligosaccharide leaven, and we have some glucose that will be generated and released. So that's why we're not changing the sugar content, right?
You're converting sucrose into glucose.
Yes.
Yes, right.
We're not changing it, but we have also the leaven. So when we do look at the calorie content, it will decrease. In addition, we have those prebiotics that can be added, but the level can be controlled. Absolutely.
Very good. Thank you.
Pleasure.
So now I don't even know if this is a good question or not, but I'll ask it all the same. So you put these prebiotic sugars, I guess is what you're saying, attach it to other sugars. Is that something that could be done for maybe pharmaceuticals? Can you attach these prebiotics to pharmaceutical compounds?
So the prebiotics to be able to label an ingredient as it has to fulfill a certain criteria, not be able to digestible, has to reach the colon, so that's I will say the two one, because it should not be digested on the top and has to reach the colon. Now, for pharmaceutical or nutraceutical uses, there are already some component that's being sold as medicine for some people that they have some, infections, they have problem of digestion, they have, they have been already, so they play the same role, but they already exist. So lactulose, like an example. So it has been used as a medicine, if you want. But what is very interesting that I couldn't talk about it today because it's still confidential, we did some animal study of our molecules to see how we can control the hypertension, and hypertension, and we have done it as well for obese people, and we found that they are able also to mitigate that and to help to reduce this to prevent those diseases. So the microbiota or our microbiome, it's so complex, right? And there are many activities there. It's not a direct effect, but many scientists they think that there is a lot of indirect effect that probably in the future, if we're able to modulate the activity of the microbiota, it will help us to limit or to prevent or even to, I will not say cure, but at least to limit really to prevent in an efficient way. Because when we look at people that they suffer from obesity, their microbiota, they have some bacteria that they are missing. Now, shall I give to this bacteria the appropriate food, then it can be peripheralated and we can have them more, and maybe this will limit the obesity. So there is a lot of hypotheses, and there is a lot of very nice and research going on to understand the activity of the microbiota. What it makes it complex, it is the fact that it is very heterogeneous. Your microbiota mind is not the same, the dynamic, all that adds complexity.
So I think I read somewhere that because of the microbiome and the benefits, that being sanitizing too much could actually be a bad thing. Do you? I've read that somewhere that. Yeah, I can't remember the exact thing, but what are your thoughts on?
So, our skin has a microbiota. And there are some now, I think, companies that they try to feed our skin with the substrates that can stimulate the good bacteria. So it's really, when we talk about microbiota, it's really our skin, our colon, it's part. And of course, if I take the example of our skin, depending on how you treat your skin, who is coming close to you, all that will affect the dynamic and the type of bacteria. And I presume the same thing applies to our colon, like what you eat each day, what your habits, your way, all this can have an impact on your microbiota or your immune system as well.
Okay, very good. Any other hands? Let's see. All right, don't see any other hands or any other questions, but this is really very interesting work. Do you have opportunities for undergraduates to do work with you, for example, during the summer sessions and stuff?
Absolutely, yeah. So we do welcome many undergraduate students during the summer from different university.
Mhm.
Yes, we have this ability to welcome undergrad as a summer student if
Mhm.
they're interested in taking part of it. So maybe I believe in just Google my name, you find the email and send me an email,
Oh,
right?
yes, 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 we do have your contact for sure.
Yeah.
All right, so, well, thank you so very much for a very interesting talk, and we hope you continued success,
Thank you very much. Thank you for
And-
having me. And
including commercialization of your work.
thank you.
All right,
Thank you.
Okay.
Bye-bye.
Okay, take care, have a good one. All right, bye-bye.